In this lecture, I'm going to review the basics of photosynthesis. If you're looking for a lecture that goes over all of the minute details, this may not be the one. But this is geared to understanding the big picture in the energy producing type of reactions in photosynthesis. And I say energy producing because I know that we're going to take the energy of our surroundings, specifically from the sun's photons, and we're going to concentrate it into a small area. And when I think that way, I think that my entropy, my change of entropy, which is denoted by delta S of the universe, is decreasing or going down in this process. Why? Well, I'm taking photons of light hitting and scattering on my leaf, and I'm concentrating all that energy, okay, into a molecule called glucose. These are gas molecules for the most part, and these are liquid, and I'm creating a solid. So just by gases have tremendous energy, liquids, and going to a solid, clearly the change of delta S in this type of reaction is decreasing. Think of entropy as the random, basically think of entropy as the distribution of energy. And if we're going from photons of light, small little particles, if you want to think photons of light, hitting a leaf, okay, and producing from that leaf, okay, this concentrated chemical energy, okay, we're gathering all this photons of light and we're concentrating it into a small area. We would see the entropy of the energy or the entropy of the universe is going down because we're gathering all the energy from the surroundings and putting it into a small little molecule, relatively speaking, compared to the size of the universe, okay? And that's glucose. So we're creating chemical energy from solar energy. So we're not creating energy, as I may have misspoke there, but what we're doing is we're transferring it. And more importantly, we're concentrating it. And when you concentrate energy, okay, we're going against the universe's entropy. Now we're going against the universe in terms of the second law of thermodynamics because energy likes to flow a certain way. Clearly, if you've got a lot of energy in a small area, like hot air, it's going to want to what? Spread out. And that's why heat flows from hot to cold. Energy that's concentrated wants to what? Spread out and expand its horizon, so to speak, by and that's why we know that energy flows in a certain direction. And that's why we know that, generally speaking, energy flows from a system of high concentration to low. And we would say the delta S of the universe increases, and that's favorable. This is a system where we're going against the law of thermodynamics that suggests, easy for me to say, that the energy disperses from high concentration. Here we're taking energy from low concentration, let's say from the total leaf, into a small concentration. So it's clear that in order to make this happen, we need energy to make this happen. As I always like to say, if I have a rock on the bottom of the hill and I want to push it to the top of the hill, I need energy or work to do so. And in this case, delta S of the universe is decreasing because I have to add energy to make this happen. Now, as we go through this process, it's always good to think about cellular respiration, which is going to have some similarities in this process, but the reaction is opposite. In cellular respiration, we're starting with a very concentrated particle of energy, glucose. It has a lot of potential energy stored in its chemical bonds. And basically, we're oxidizing it, or we're releasing that energy that's stored and using that energy to make ATP. And when we start with high concentration of energy into low, delta S of the universe is increasing. So this reverse reaction, cellular respiration, would be S if we already have this rock on top of the hill and we're not applying any work on it. It's what? Going to roll down the hill. Kind of like if I'm riding a pony. If a pony is making me move somewhere, okay, I've got free energy. The pony is giving me the free energy to move. So the free energy, we would say delta G, is negative when I go in this reverse process, cellular respiration, right? And delta S of the universe is increasing. That's me coming down the hill. But in photosynthesis going in the forward, the delta S is decreasing and the delta G is positive. All of these give me the reasoning here that I have to add energy to make this happen. I have to fight the universe's flow of energy to take random bits of energy, photons of light, and concentrate it into this chemical energy of the glucose. And of course, this is what life is all about, taking the energy from the sun and converting it for a usable form for other animals. Obviously, the autotrophs, okay, of our planet do this, okay, and then of course the heterotrophs, the consumers, okay, can use this in cellular respiration. Don't get lost in the fact that the plant cell itself also undergoes, okay, cellular respiration as well. So it's important that we get that. Now, so here we are dealing with the forward reaction, so let's not confuse ourselves with cellular respiration here. So we have a process by which we need energy to concentrate this energy, and it's again a process where the delta G is positive, delta S of the universe is decreasing. So if I'm on that pony and I want to move, I don't have free energy. I've got to get off the pony, pick him up, and carry him to move somewhere. But thank gosh we have a constant supply of energy to do so, unlike me carrying the pony, which is the what? The energy of the sun, the photons of the sun. So that's really important as a background as we go forward in all of this. All right, so what is that endosymbiotic type of cell that we deal with? And that's called a chloroplast. As we've learned, at some point in evolutionary history, maybe cyanobacteria was engulfed by a prokaryote that was now able to, in symbiosis, be able to help each other work. In fact, the chloroplast, which is probably the cyanobacteria that was engulfed in evolutionary times, okay, at some point, probably 2.5 or 3 billion years ago, helped prokaryotes produce energy, okay? And in the chloroplast, we have these discs called thylakoids, and they have also membranes, kind of like, a lot like the mitochondria, okay? So let's take a closer look at what's going on in this system here. So when we talk about photosynthesis, we're talking about two types of reactions. You'll hear light and dark reactions, okay? And we're going to spend our first part of this in the light reactions. Now, the light reactions just mean that we need the sun's energy to do so. Now, there's two parts to this. There's the light reactions, and then we have the dark reactions. And again, that just means that we don't need the light to make this work, okay? Both reactions will occur in the presence of sunlight. Obviously, the light reactions do not. Now, where do the light reactions occur? Well, this occurs in the thylakoid, in the chloroplast. They're all both in the chloroplast, but specifically in the thylakoid. Okay? Now that's an A there, so the thylakoid. Now these are these discs, okay? These are basically discs that stack up on themselves, and you can see them in a electron microscope because you really can't see them with light microscopes. But they do disc up. They do basically stack up on each other, and so we're looking at one thylakoid, and that's where the action is, and that's what my diagram is above. Now, in the light reactions, it's important you understand that we're going to basically harness the sun's energy, solar energy, and from that, we're going to make two things. Okay, one is we're going to make ATP. When you may say, why are we making ATP? Why that? Well, we're making ATP because we need energy to push the rock or the pony up the hill. Okay, this is not an exergonic reaction. This is a reaction where we need energy to force this to happen. So we need to make ATP. That's the first thing. The second thing the sun's going to give us in these light reactions, it's going to give us, okay, NADPH. Now what this does for us, this is going to be a high energy electron carrier. It's going to hold 2 electrons, and we're going to need these high energy electrons to reduce the CO2. Now, these are redox reactions, and let's review what we're talking about here. If I go back to my original equation, so when we look at CO2, carbon, the Lewis dot diagram looks something like this, where I have 2 pairs of electrons shared, that's a double bond, and then 2 lone pairs, and then I have the same thing over here. And you had done this in chemistry. Okay, and what it really is it's a carbon double bonded to an O, double bonded to another O. Okay, and it's got that strong double bonds that keep it very stable, which is important because we want the CO2 to get back to the leaf, right? After we use up the O2 in cellular respiration, the product of that is CO2, which we want to reconvert back to oxygen so we can continue to breathe. That's why this photosynthesis cellular respiration cycle is so very important. But back to CO2. Because oxygen is so very electronegative, it pulls these pairs of electrons away from it, which means this carbon sort of does not have these electrons. Why is that important? Well, we want to bring it back to the glucose, which is going to have a carbon to hydrogen bond. Now, a carbon to hydrogen bond is almost shared equally down the middle. We call this essentially a non-polar bond. And these electrons are high energy electrons. Why are they high energy electrons? Because they're more freer to move. Here, these electrons are pulled closer to the oxygen. These are lower energy electrons. And essentially, that's what we're doing in photosynthesis. We're changing these highly bound electrons that are being pulled closer to the oxygen and now letting them be freer in the carbon-hydrogen bond of glucose. To go from low energy to high energy, guess what? We need energy. And again, that's why it's an endergonic process. Now, in order for carbon to what? Regain these high energy electrons or kind of feel like it has them now. Here it feels like it lost them, notice the space, and here it kind of has them. CO2 needs to be reduced, or the carbon in CO2 is reduced. And the reduction is really important. In the opposite direction, we oxidize the carbon. That's what burning is. Okay, so it's very important that reduction has to occur here. Now, reduction, if you remember your redox, Leo the lion says gur, oops, in reduction, okay, in reduction, we're gaining electrons. And because we're gaining electrons that are negative, our charge gets reduced. But this carbon has to regain these electrons, so therefore they're closer to it. And if they're in the middle of these two, they're more higher energy. So it's important that you realize the reduction here. The water gets oxidized because you can't have one without the other. In the reverse, okay, again, it's always important to think about this process in reverse of cellular respiration. It's the sugar or the glucose that gets oxidized. All right, and again, we want to create these high energy electrons. So reduction is occurring while the water gets oxidized. And in the other reaction, the glucose gets oxidized in the reverse and the oxygen gets reduced. But it's important that you see that the reduction is occurring here for the carbon. That's really important. And that occurs, okay, in the second part. But again, the light reactions that occur, all right, in the thylakoid membrane, okay, mostly in the membrane, we're producing ATP, which we need to force the rock up the hill, but more importantly, probably, is to produce, okay, this reducing agent, or this NADPH, that takes these high energy electrons that are generated from the sun and carries them into the dark reaction, which does not occur in the thylakoid. It occurs in the stroma outside the thylakoid, okay? And we take this ATP that's made and these high energy electrons that we're going to use to reduce the carbon, and we call that reduction of the carbon, or at least adding the CO2 together, we call that carbon fixation. I want to add the carbon together. So you can see that the CO2 that is being absorbed through the stromata, the holes that are guarded by the guard cells into the spongy layer of the leaf plant, is used to fix and add the carbon together. And once it's added together, we reduce the carbon that are put together so that it can have a carbon to hydrogen bond. All right. So the first part, the light reactions, is creating ATP and these high energy electrons so that the stroma, the outside of the thylakoid, does not lead light. It uses the ATP and the high energy electrons that the NADPH is carrying from the light reactions into the stroma to force the rock up to hill, or to basically concentrate the energy, okay? But to do so, we've got to take the CO2 and fix it, put it together, add it together, and then reduce it so that we create these high energy bonds in glucose. Okay? Now, let's talk about a little more specifics in the process. So during the light reactions, as you can see, we have two photosystems. Okay, we have photosystem 2 and photosystem 1. It's interesting that they actually discovered or named this one, or discovered it first. But this is the first part. Now, in a photosystem 1, you have to understand that what we're dealing with is these reaction complexes. Now, we have these light harvesting complexes that are basically here, okay? And what they do is they collectively on the membrane, notice this is a membrane where it's a bimembrane, right? Biphospholipid membrane here. This absorbs photons of light in a certain region. Now, what these light harvesting complexes are made up of are proteins, chlorophyll, the carotenoids, a bunch of different pigments that collectively absorb a wider length of light than without these proteins or without these other pigments. So these pigments that grab a wide range of light. Now, you see these little green circles here, okay? These represent those different kind of pigments or chlorophyll that get excited. So what happens is light comes in, as we talked about. We have electrons that are closer to the nucleus that are stable and low energy. Light comes in, pushes these electrons to a higher energy state. Now, these higher electrons will jump back down, okay? And sometimes this emission jump back down gives off the energy needed to excite other electrons up, and that's what happens. So light hits these, let's say, chlorophyll embedded with these protons and carotenoids, and it excites them. It makes them jump to higher energy levels, okay? And when they do so, the ones that are next to it are basically excited as well. So as we have electrons that jump farther away from a nucleus, that positive nucleus, and get excited, when they jump back down, they release energy that can what? That energy is used and absorbed by the next little green circle, or light harvesting complex. And what that does is take electrons that are close to the nucleus, and they get excited. When this high energy electrons jumps back to get stable, that energy is being used to excite the others. So if I was to redraw this complex, this light harvesting complexes, okay, they're next to each other, and what they do in a chain reaction, so this one absorbs energy, this gets excited, this releases the energy as the electron jumps back down, this gets excited, when it jumps back down, the electron releases energy, and so forth and so on. So you can see that the energy absorption, excitation, and release of energy forms a chain reaction that basically works its way into this pair of chlorophyll A's. And we call these basically the reaction center complex in the middle. All right, and these pair of chlorophylls, okay, along with some enzymes, basically pull electrons away from the water. So you can see the water here is being metabolized. And what happens is water, as we remember, has two pairs of electrons with each hydrogen and has two lone pairs here. And what happens is in this process, in this reaction center complex, there's an electron acceptor in right here. And an electron has to be transferred here. So the energy travels through the light harvesting complexes into the reaction center complex. And then there's a double molecule chlorophyll A here that takes that energy and essentially rips electrons away from it into this complex. So electrons are basically removed here in a redox reaction where electrons are lost. So this light energy rips electrons away. Now, electrons are being ripped away here. Where do they come from? Well, they get reabsorbed by the water. The water provides the electrons. So as we pull electrons away from the water molecule, and they're pulled away from these hydrogen bonds, so to speak, from the water, these H's leave as H pluses, and they increase the concentration of protons in the thylakoid membrane. They contribute to that, which is what we need, and I'll explain that in a second. So what's left is an oxygen with 6 electrons, and these 2 electrons are replenished right here. And again, with the energy, okay, these electrons get ripped off into this electron carrier, okay, or electron, I should say, acceptor. And from here, they're able to move through an electron transport system. Now, before we go through that, what happens to these oxygens? Well, this oxygen, who has 6 valence electrons, will hit another oxygen who has 6 valence electrons. And as we've learned, what happens here is that we create an O2 molecule with a double bond. Each of these feel like they have the 8. And by gosh, the O2 is, I don't know why people always do that, is the what? A byproduct of this process. So we produce and replenish the earth with oxygen, okay, that we need to breathe in order for cellular respiration or aerobic cellular respiration to occur. So it's a really important part of photosynthesis, although it's not an important part in this process to build the sugar. All right. So what happens is this high energy electron goes down through these protons and cytochromes and proteins, and basically using its high energy, okay, it helps pump protons from the what? Stroma outside this membrane-based thylakoid and pumps the H pluses into it. So we're building a proton gradient as this electron is passed from one protein cytochrome complex to another. This electron transport chain is a lot like what happens in the mitochondria. So basically, we're pumping protons through this system. We're using the energy of the electrons to pump protons, and we build up the protons in the thylakoid membrane, okay? And this buildup of protons creates a gradient. In fact, the pH in this membrane becomes something of a 5, if you remember. And the pH outside this is about a pH of 8 when there's light. And if you remember, a pH of 8 means I have a concentration of protons of 10 to the negative 8. And in here I have a concentration of 10 to the negative 5. So you would say that the H plus concentration is 1,000 times greater inside the thylakoid space here, okay? Remember, 10 to the 5 is 1,000 times bigger than 10 to the negative 8. Actually, 10 to the negative 5 is 1,000 times bigger than 10 to the negative 8. And that's very important to understand. Okay? And that 1,000-time greater concentration makes these H pluses in this gradient want to rush out. And of course, here is our ATP synthase, just like we have in our mitochondria, that allows the H plus to rush out. And by rushing out, turning this turbine, okay, creates the energy to make ADP into ATP. And this is where we create the ATP that's necessary to force the rock up the hill and make the sugar, the concentrated form of energy. Okay, so that's important. Now, once this high energy electron helps produce or pump the H plus, it kind of loses some energy, but not all of it. And this semi-lost high energy electron, if you want to call, is now used for the photosynthesis 1. And the idea here is that this works the same way, except it doesn't take the water, it takes the electron that already has some stored energy in it, because not all the energy was lost to help pump the protons in. So this is coming into this photosystem that's essentially identical, okay, to photosystem 2, and this higher energy electron that's not completely lost its energy gets taken up into this chlorophyll A molecule, or these double molecules. They call them P780s or 700s, but not really important to know that detail. But we have this electron, it didn't lose all of its energy being taken up. And what's really cool is that this energy, or this higher energy electron, okay, absorbs very similar wavelengths of light here, but now we're able to push this electron into higher energy levels because it's coming in with some energy already, unlike the scenario here. And that same light that it absorbs, and we've talked about what region of light, okay, in the red and blue spectrum, mostly the red here, is able to excite this electron that already has some energy into, even into higher energy levels. So it's removed by the electron acceptor, and it has even higher energy. And with an electron carrier and an enzyme, okay, and the enzyme's name is pretty cool. It's called NADP+ reductase. Kind of gives you the idea that this is going to be used to help reduce, help push, or give back those electrons to the carbon, as we talked about. So through this enzyme, okay, we make NADPH. So NAD+ is able to gain actually, okay, 2 electrons, okay, and a proton, all right? And that is channeled into the Calvin cycle outside the thylakoids. And now we have our ATP, and now we have our NADPH, which is the essentially the stored high energy electron. And notice how we made that high energy electron. We needed that electron to be somewhat charged already in order to make it jump into higher energy positions. Because both of these photons of light being absorbed are the same. So the same amount of energy is going in, but if we're starting with an electron that already has some energy already, it supercharges that electron into higher energy levels that the NADP plus can hold on to until we need it to reduce and free up those electrons in the Calvin cycle. So now we've set the stage. We have the light reactions producing the energy, ATP, that we need to climb up the hill, to concentrate the energy, to go against nature. And we have the electrons that we need to reduce, okay, the high energy electrons we need to reduce the fixated carbon that's building in the Calvin cycle. All right. And now let's go into the Calvin cycle. But again, I'm not here for the I'm not here to talk about all the basics or all the details, just the basics. Again, the light reactions in the thylakoid, okay, produce the ATP that we need, and it produces the reducing agent, the agent that helps reduce these compounds to free up the electrons. So the first thing, we take the CO2, and normally the CO2, notice it does not go into the thylakoid. It's not part of the process. The CO2 is outside the thylakoid in the chloroplast. And this CO2, with one of the most famous of all, okay, our enzymes, Ribisco, talked about a lot as the most abundant protein in the earth, fixates or adds this 1 carbon system to an already 5 carbon, okay, These grays are 5 carbons that are being cycled in the Calvin cycle. So now we've got a 6-carbon system because we've just fixated or added a carbon, which is unstable. Immediately it splits, okay, into 2 3-carbons. And then we add some ATP to add the, what? To phosphorylate them, to give it the energy, and then we reduce it with our high energy what? High energy electrons. And what we produce essentially in this process is a 3-carbon, they call it G3P, okay, but no worries about it, a 3-carbon hydrocarbon that has the carbon bonded to the H in this way that the electrons are loosely held, and we've made the high energy electrons. So with the use of an enzyme, bunch of ATP, okay, and of course high energy electrons, we've created a 3-carbon precursor, because as you should know, we don't make glucose directly. We can make glycerol, we can make glycogen, which is a starch, we can make a couple of other different things besides glucose in this process. But the bottom line is all of these have these carbon to hydrogen bonds that are loosely held. Don't forget, CO2 had these electrons kind of pulled away from it. And we wanted to create a scenario where the carbon doesn't really have these free electrons to a scenario where we do. And that's where the Calvin cycle comes into play. And of course, it has to, because we're adding 1 carbon per cycle here, this thing has to go 3 times in order to make 1 3-carbon sugar. Okay, so you can think how many times it has to go to make glucose, right? It would have to go 3 times to make a 3-carbon sugar, so you need 6 times to make total 1 glucose. So you can see the energy involved. Need the ATP, we need the reducing agent, or the one that's carrying the high energy electrons, okay? But the bottom line here is that the light producing reactions, okay, the light producing reactions use the solar energy, okay, to generate the ATP and the NADPH to provide the chemical energy, okay, and the reducing power to make this work. All right, so big picture is important. Certainly there, for those looking for more details in this process, okay, this is about understanding that the big picture here, okay? So that's photosynthesis, and I hope you grab some of the basics from this.